Hello everybody, you are welcome. Today we will solve together paper 23 October-November 24 Cambridge AS Biology. In this exam we will partially focus on certain questions, certain topics. In question number 1, gas exchange and heart systole and diastole. In question number 2, mitotic cell cycle. In question number 3, nucleic acids. Number 4, biological molecules. Number 5, immunity. Number 6, cholera. Let's start the first question. Number 1, figure 1.1 shows the structure of the human gas exchange system with letters from A up to G. Okay, here's a question about use the letters in figure 1.1 to identify the parts of the gas exchange system that contain cartilage. Cartilage is found in these structures in letter A, larynx, also found in trachea, okay, plus it present in C, bronchus. Okay, what's the difference in the cartilage shape in both trachea and bronchus? In trachea, it has C-shaped structure in complete rings. In bronchus, it represented by irregular blocks of cartilage. Okay. Now, this answers. Okay, B and C, accept A also. Okay. Double I describes the role of cartilage in the gas exchange system. What is role? Here, look this trachea, and behind and parallel to trachea esophagus, it lies parallel behind the trachea, okay, at the back of trachea. Okay, here what the importance or the role of cartilage in the gas exchange system, either in trachea or larynx or even in bronchus or bronchi. It prevents collapse of air pathway during inhalation and exhalation. This is number 1. Also, it allows flexibility, bending our neck, swallowing food, where esophagus make peristalsis motion during swallow. This allows freedom of esophagus to move by peristalsis. Okay. C-shaped cartilage rings allow widening during breathing, during inhalation and exhalation, according to depth of breath. Okay, according to depth of breath. Now, triple I. Microscope slides were prepared from 2 regions of the gas exchange system. Here 2 slides. Okay, photomicrographs of the 2 slides. Figure 1.2 with magnification 120x. And the figure 1.3 with magnification 560 times. Okay. The question here, complete table 1.1 by focus on 3 bullets. Use the letters from figure 1.1 to identify the regions. Where is figure 1.1? Here we asked to use the letters from A to G. Okay, to identify the regions of the gas exchange system from which the 2 slides were prepared. These 2 slides, figure 1.2, okay, and figure 1.3, from which letters here in this figure. Okay, identify 1 features visible in figure 1.2 and 1 feature visible in figure 1.3. Then state 1 way in which each feature relates its function, its adaptation, or its function related to its A function. Okay, here look, this is the table 1.1. Here as the left column, figure 1.2, region from the gas exchange system from A to G, 1 visible feature, 1 way in which the feature relates to its function. Look here, figure 1.2 and figure 1.3. Also, figure 1.1 is a table again from figure 1.2. From figure 1.2. From which organ we made this slides? From which organ? From this shape. From where? Okay, exactly here, air spaces, a lot of air sacs, alveoli, so the letter G, the answer letter G. Here region A to G, G, one visible feature, air spaces, yes. One way in which the feature relates to its function, large surface area for gas exchange. A lot of air spaces, this create large surface area for gas exchange. Also, we note that in figure 1.2, alveoli characterized by thin walls. Why? To create less diffusion distance and faster rate of diffusion. Thin walls, this creates short diffusion distance and also increase diffusion rate. Plus capillaries. Where is capillaries in this figure? Look, capillaries in this figure 1.2 here in this black areas, in these corners, okay, surrounding alveolar wall. Okay, all this capillaries, a lot of capillaries. This maintains steep concentration gradient between gases, both gases, oxygen and the carbon dioxide, even in air inside alveoli and blood inside blood capillaries. This maintains steep concentration gradient. Okay, you can choose one of these three features. What about figure 1.3? From which organ we made this slide? Which organ from this figure? Here, goblet cells, a lot of goblet cells, okay, and ciliated epithelial tissue. From which organ we made this slide? Yes, right, from trachea, good, or even from bronchus also. Plus A, plus larynx. So any letter from this is good answer for the region. Look here, 1.3 made from organ B, trachea, except C and A. C and A also are correct. One visible feature, goblet cells that secrete mucus, okay, helps stick germs or pathogen. Okay, what else? Ciliated epithelium, ciliated epithelial tissue moves mucus towards the throat. The cilia can move, can waft, moving the mucus with dirt, with germs towards the throat. It's okay? Okay. Now for letter B. There is a regular and efficient supply of blood from the heart to the lungs. Describes the sequence of events that occurs in the heart to make sure that there is a regular and efficient supply of blood to the lungs. Here we asked to discuss or describe the sequence of events that occur in the heart to make sure that there is regular and efficient supply of blood to the lungs. Okay, blood passes from the heart to lungs. From which part? Left or right? From right side. Okay, from right side, from right ventricle. What is the mechanism of events or sequence of events that helps regulation and efficient supply of blood to the lungs? Look, step by step. Here in this figure, we revise mechanism of heart beating. Mechanism of heart beating. As we know, the heart is myogenic in action. Automatically, by its own, it pumps blood, okay, away from the heart itself, away from it, by pumping blood away in arteries. How come? By certain fibers located as a right, buried in the right atrium wall. It is called sinoatrial node, SAN. Sinoatrial node, SAN. Okay, this releases impulses, electrical signals that spread in both atrial wall. This creates or allows both atria to contract at the same time. This called atrial systole. Allow blood to pass down through these valves, AV valves, to both ventricles. Okay, here we concentrate on the right side, on this area on the right side. Why? Because he asked about from heart to lungs, from heart to lungs. So we concentrate the journey of blood from the heart to lungs in this side. Then what about this signals after reaching both atria, after atrial systole, after reach blood to both ventricle or right ventricle? The blood in the right ventricle stay till another signal coming from where? From AV node, atrioventricular node. Okay, this delay or relay the signal where it passes it only through the septum, through Purkinje fiber or Purkinje fibers that passes down in the septum to reach the apex of the heart, then to the wall of both ventricles, right and left. This allows both ventricles to contract this ventricular systole. Blood passes from right ventricle towards the pulmonary artery, okay, by the help of opening of semilunar valve. Look, I have to remove this or erase all ink. Okay, then we concentrate step by step. Again, here sinoatrial node SAN generates impulses. Yes, impulses spread across the atrial walls. That's just period for atrial systole. Look, right atrium contracts this atrial systole. Tricuspid valve opens. Tricuspid valve open, allow the blood move into the right ventricle. Here atrioventricular node AVN sends impulses down Purkinje fiber, down Purkinje tissue, through Purkinje tissue in the septum. AV node introduces time delay, relay. Okay, then right ventricle contracts this ventricular systole. Semilunar valve opens and blood flows into the pulmonary artery to reach the lungs where gas exchange takes place. Here tricuspid valve, after blood pumped in a pulmonary artery, tricuspid valve closes to prevent backflow, and also semilunar valve closes to prevent backflow also towards the right ventricle again. Look at this mechanism. Concentrate for several seconds. This fully match with the discussion on the left side. Okay, now question number 2. Stem cells are found throughout the human body. LGR5+ stem cells are found in the lining of small intestine. Figure 2.1 is flow chart showing stages in the development of one of the daughter cells produced by mitotic division of an LGR5+ stem cells. Explain why stem cells are required in places such as the lining of small intestine. Small intestine, where foodstuffs fully digested, okay, where completion of digestion takes place by the last steps of enzymatic reactions. So this cells subjected to enzymes, subjected to chemicals and foodstuffs. What happens to the cells? The cells aged, the cells died, okay, and must regenerated. Okay, so stem cells present in the lining of the small intestine. They are required to regenerate, okay, missed or compensate missed or lost cells due to digestion. Okay, here the answer now: the stem cells doing mitosis in certain stages to reach to be specialized like this 4 cell types. Specialized cells from epithelium lining the small intestine, from where? From unspecialized cells called stem cells, LGR5+. It's okay. Here, why is the cells present in the lining of small intestine? To replace worn out cells and repair tissue. Also, for stem cells divide and differentiate. Stem cells able to divide continuously without stop. Okay, they are undifferentiated and help to produce differentiated cells like this 4 cells. Okay, now for double I, name the stage of mitosis shown in cell X. What's the stage of mitosis here in cell X? Yes, here group of chromatids and group of another chromatids, sister chromatids. They are separated now from each other. Okay, one move to the left, one move to the right. Okay, so this called anaphase, where chromatids splits and move away from each other. This phase called anaphase. Okay, anaphase. Now for triple I, state the part of the cell cycle shown at Y in figure 2.1. What's the name of Y? Stage or part of cell cycle. Cell cycle divided into 3 parts: interphase, mitosis, and last one, cytokinesis. Cytokinesis. Look at cytokinesis, please. After nuclear division, the cytoplasm must be divided into 2 parts, into 2 daughter cells. This is called cytokinesis. Again, shrink of cytoplasm. Okay, like this. Again, last time, here after telophase, cytoplasm divide into 2 portions, into 2 daughter cells. This is called cytokinesis. So Y is called cytokinesis. Okay, now for P, explain the role of centromeres in the cell cycle. Explain the role of centromeres in the cell cycle. Centromere here join both sister chromatids together. Okay, what is rule? Sister chromatids remain attached, okay, by centromere. Chromatids attached to spindle fibers. Centromeres are the sites where spindle fibers attach to certain part called kinetochore, just near or very close to each centromere, called kinetochore. Chromatids attached to spindle fiber. Also, centromeres divide in anaphase. At end of anaphase, the centromere split or divide into 2, where each chromatid, sister chromatids, contain one of them. Okay, that allows spindle fiber to detach group of chromatids from other sister chromatids on the other side. Here, chromatids move to opposite poles by help of the centromeres. Letter C, figure 2.2 shows 3 types of specialized cell that develop from LGR5 plus stem cells in the small intestine. Okay, 3 types of cells. Okay, the structure features of the cell indicates its likely function. Suggest the function of each of the cells shown in figure 2.2 and explain how the structure of each cell supports your suggestion. Look here, this cell What about this cell? Its function. It possesses, it have at its free end a lot of microvilli, a lot of mitochondria. Okay, rough endoplasmic reticulum, mitochondria. Okay, look please. Letter B, absorption of nutrients. This microvilli increase the surface area needed for absorption. Absorption takes place at microvilli. Also digestion, completion of digestion, where disaccharidases like sucrase, like lactase enzymes found in the membrane of microvilli. Okay, this immobilized enzymes found in the membrane of microvilli. Microvilli increase surface area. This structure adaptation, microvilli increase surface area a lot. Cell Q. Cell Q, goblet cell, contain a lot of vesicles containing mucus. Goblet cells contain vesicles for secretion, for mucus secretion. It's okay, this for cell letter Q. Last cell, R. R contains extensive network of rough endoplasmic reticulum. So this cell considered highly secretory cells. It produces enzymes for digestion. Okay, in vesicles, a lot of vesicles, this migrating or transport vesicles reach the cell membrane boundary to release its content of enzymes. This secretory, so enzyme, it is needed for enzyme production. Structure adaptation, it contains a lot of rough endoplasmic reticulum to produce enzymes. Okay, and the Golgi apparatus for modification of these enzymes. Okay, and a lot of vesicles for exocytosis, needed for exocytosis as last step of secretion. Question number 3. The 5 bases found in nucleic acids are described as nitrogenous organic compounds. There are 2 types of base. Figure 3.1 shows the structure of the 5 bases: uracil, thymine, cytosine, adenine, guanine. Okay, the question here, state the name of the type of base that includes U, T, and C. Name of type of base that includes U, T, and C. What's the name? What's the family name of chemicals represented by U, T, and C? Only one ring, one nitrogenous ring. That's called pyrimidines. Pyrimidines. Okay? Pyrimidines. Now, to revise nitrogenous bases. Nitrogenous bases in DNA or RNA divided into 2 groups. Pyrimidines with one ring only. Okay, cytosine, thymine, uracil, and other nitrogenous bases with 2 nitrogenous rings, like here purine, guanine, and another purine, adenine. A and G from purines. This is called purines. Okay, family of purines. Okay, to revise, look please, any one of these 5 nitrogenous bases, okay, it join with carbon number 1 in pentose sugar, okay, and the carbon number 5 of this pentose sugar is joined with phosphate group. So the nucleotide is formed from 3 parts: sugar, pentose sugar, nitrogenous base attached to carbon number 1, and phosphate group attached to carbon number 5 of this pentose sugar. Here, what about this pentose sugar? Either its name may be deoxyribose or ribose according to the DNA or RNA. In RNA, the sugar is ribose, where carbon number 2 and the carbon number 3 both attached to OH group, OH, carbon number 2 and 3, while in deoxyribose, carbon number 2 is devoid or is lack of one oxygen, so it is called deoxyribose sugar. It's okay. This is just revision of nucleotides of both DNA and RNA. Now look, state why it is not correct to say that all nucleic acids have 5 bases. All nucleic acids, we mean DNA and RNA. Each one of them only consists of 4 bases. For example, look please here, for DNA deoxyribonucleic acids, the 4 bases: thymine T, cytosine C, adenine A, guanine G, A, G, C, T. Okay, while in single-stranded RNA or RNA that form single strand only, okay, that's formed from single strand, only 4 bases: uracil instead of thymine, while the other 3 nitrogenous bases are the same like DNA. Okay, here, so it is not correct to say that all nucleic acids have 5 bases, only 4 bases: A, T, G, C in DNA, U, A, C, G in RNA. So the answer, each nucleic acid has only 4 bases. DNA has thymine T, while RNA has uracil U. It's okay. Now for letter B, figure 3.2 shows stage in the replication of DNA. The circle part is enlarged. The circle part is enlarged in figure 3.3 to show the elongation of the DNA strand that is being synthesized. Look as magnification. Okay, this magnified circle part in figure 3.3 now. Okay, the question here: describe the sequence of events that occurs at the stage shown in figure 3.3 to extend the synthesized strand. Okay, synthesized strands, I mean here from 5 to 3 dash, from 5 direction to 3. This for the new strands, newly synthesized strand. What the events, sequence of events? Okay, for 4 marks. Activated nucleotides, from where? Here, this activated. Each nucleotide only contain phosphate group, 1 phosphate group. Here 2 extra phosphate group. So this activated nucleotides, okay, pair with complementary bases of the original or template strand. This is called template strand or complementary strand. Okay, activated nucleotides pair with complementary bases. Thymine pairs with adenine. Thymine paired with adenine. Adenine from purine. With double rings from here, adenine. Okay, and thymine, this coming nucleotide. Okay, thymine with from pyrimidine. Okay, with only 1 nitrogenous ring. So thymine pairs with adenine. How this pair takes place? By forming hydrogen bonds. How many hydrogen bonds between thymine and adenine, or A and T? 2 hydrogen bonds. Okay, DNA polymerase catalyzes phosphodiester bond. After pairing, certain enzyme, essential enzyme called DNA polymerase, its good direction is 5 to 3. Okay, join this nucleotide. Look at this nucleotide, previous nucleotide with next one, with this one. How come DNA polymerase catalyzes phosphodiester bond? New strand elongates from 5 to 3. Now extra nucleotide is added in the direction 5 to 3. Look at phosphodiester bond, how it is formed by DNA polymerase enzyme. Look please, this already attached this newcomer nucleotide. Okay, after base pairing with another strand by 2 hydrogen bond, okay, like here for example, here with thymine, okay, here with adenine for example. Now how we join this last nucleotide is a newly synthesized strand and with newcomer by forming what? Phosphodiester bond. With removal of water, this is considered condensation reaction because water is released. Okay, and here phosphodiester bond is formed, C, okay, from previous nucleotide, and O, it's O here, okay, join with phosphate group of another or next nucleotide. This is done by DNA polymerase enzyme. This is called phosphodiester bond. It's okay? Okay. Look please, another figure to show phosphodiester bond. Here, this phosphodiester bond, okay, between C, O, B, okay, O, C. This is called phosphodiester bond. Here also the same, another phosphodiester bond. Okay, okay. Letter C. The two strands in a molecule of DNA are described as antiparallel with reference to figure 3.2 and figure 3.3 state what is meant by antiparallel and explain how the antiparallel arrangement of the strands determines how new strands are synthesized. As we know, DNA is called double helix. It is formed of two strands run parallel to each other but antiparallel. This means they are opposite in their direction. Look, okay, DNA strands run in opposite directions. The first strand is direction 5' end towards 3' end down, while the other one 5' end to 3' end up. They run in opposite directions. Why? To allow base pairing between adjacent nitrogenous bases, G with C with 3 hydrogen bonds, A with T with 2 hydrogen bonds. If they run in the same direction, impossible to make base pairing between these nitrogenous bases. So no rungs are formed if they are run in the same direction. Okay. Now, with the second part of the question, explain how the antiparallel arrangement of the strands determines how new strands are synthesized. Since the direction is opposite, so the replication of DNA, both strands of DNA are different in action, different in synthesis. How come? Look please at this figure. The original DNA represented here by blue strand and red strand. This is the original DNA before replication. To allow replication takes place as this two template strands, DNA helicase unzips in this direction. So the original strands become far apart each other, allow building and replication of the two strands, newly strands. In the first one here, look at this one, DNA polymerase run in the same direction like DNA helicase in this direction. DNA polymerase can add and make phosphodiester bond step by step, nucleotide by next one, to form continuous leading strand. This is the original direction of DNA polymerase, 5' to 3' of the newly synthesized strand. While, what about the second strand? DNA polymerase unable to move in opposite direction. So, DNA polymerase can make certain pieces of DNA called Okazaki fragments, like these arrows, blue arrows. Certain segments, not complete, not continuous. This is called lagging strand. Okay? Then DNA ligase make phosphodiester bond between this Okazaki fragments to form newly synthesized strand of the other template. It's okay? So the answer, now DNA polymerase moves in the 5-3- direction, is the correct direction. Leading strand is synthesized continuously, while lagging strand in fragments, Okazaki fragments. Okazaki fragments are joined by DNA ligase. It's okay? Okay. Question number 4. Biofuels contain alcohols that are produced by the fermentation of sugars derived from the crop waste. This waste contains cellulose and other organic compounds in cell walls. Scientists investigated the production of sugars from crop waste for biofuel production. The scientists discovered that a strain of the fungus Penicillium citrinum, isolated from soil, was a good source of 3 different extracellular enzymes, M, N, and O. These enzymes break down polysaccharides in cell walls. The scientists cultured B. citrinum in liquid medium containing cell wall material. Samples of the liquid were taken and the 3 enzymes were separated from the medium. Each enzyme was placed in reaction mixture with an appropriate substrate. The activity of each enzyme was determined to give a measurement of the quantity of the enzyme produced by B. citrinum. Optimum conditions for each enzyme were used to obtain the results in table 4.1. Look at 4.1, enzyme M, N, and O. Maximum activity by arbitrary units, per arbitrary units. This figures, okay, 200, 292, 111, 6.5, and so on. Okay. The conditions were different for each enzyme. Okay. Optimum conditions for each enzyme were used to obtain the results in table 4.1. Look at table 4.1, the enzyme M, N, and O with maximum activity per arbitrary unit. Okay. The conditions were different for each enzyme. The scientists carried out further research so that a solution containing the 3 enzymes enzyme mixture could be used for the most efficient production of sugars from crop waste. Suggest what the scientists needed to find out in their research. This question merit 4 marks. Okay. Firstly, the scientists need to identify suitable crop waste, which waste used to create much more or most efficient production of sugar. Which waste? Which crop waste? Okay, identify suitable crop waste that create or produce more efficient amount of sugar. Then determine optimum pH and temperature for each enzyme. Each enzyme different from the other, so they behave and works in different conditions. So scientists need to determine optimum pH and temperature for these 3 enzymes. Then find the best enzyme ratio and concentration. Which enzyme with which ratio and concentration is needed to create efficient production of sugar. Okay. I mean, say for example, enzyme M. We prepare different enzyme concentration of M, at which enzyme concentration create this solution, this concentration, the biggest amount of sugar from certain waste. Okay, find the best enzyme ratio and the concentration. Last step, investigate inhibitors and cofactors to avoid inhibition, to avoid lack or deficiency in production okay, or impairment of production of sugar. We have to determine inhibitors, either competitive or non-competitive, and the cofactors that needed to activate enzymes to create best production of sugar. It's okay. Now, for letter B, students investigated the composition of the cell wall of leaf cells of Thalicrus arabidosis thaliana. The students began by isolating the cell wall components from the rest of the cell material. As we know, just this is a hint, cell wall not made only of cellulose. It made of cellulose and other chemical compounds, like what? Like hemicellulose, like lignin, like pectin as glue substance to stick cellulose molecules together. Also protein, okay, like expansion protein. Okay, the student began by isolating the cell wall components from the rest of the cell material. The student used enzymes extracted from a fungal pathogen of A. thaliana to hydrolyze the cell wall components to smaller molecules. The student prepared a reaction mixture containing the cell wall components and the enzymes. After 24 hours, they separated and identified the smaller molecules found in the reaction mixture. After hydrolysis, okay, the student separated and identified the smaller molecules found in the reaction mixture. 4 types of molecules were identified. Short chains of beta-glucose, peptides, amino acids. Explain the presence of these molecules in the reaction mixture after 24 hours of hydrolysis. Why beta-glucose is present as short chains and as single units of beta-glucose? As we know from our study, cellulose molecules is formed from beta-glucose. Okay, the enzyme able to cut the cellulose molecule into small pieces, short chains of beta-glucose, and individual units of beta-glucose also. Okay, offshore the cell wall contain protein, so why peptide is formed here in the results after 24 hours of hydrolysis? Peptides means short chains of amino acids plus amino acid itself. So the answer here, look at cellulose molecule, where here O that join carbon number 1 and the carbon number 5, okay, here in the opposite direction it rotates. Each successive sugar molecules, beta sugar molecules, rotate 180 degrees. Why? To allow joining this bond to form glycosidic bond in this form, this beta glucose units. Okay, now cellulose breaks down into beta glucose by cellulase enzyme. Proteins break into amino acids. What about partial hydrolysis, not complete? Complete form basic unit, like here in the first step, basic unit beta glucose, and here in second step, basic unit or block called amino acids. This complete hydrolysis for different chemicals, cellulose and proteins, into single units, into monomers, while partial hydrolysis create or produces peptides and short beta glucose chains. Peptides from proteins and the beta glucose from cellulose. Okay, here enzymes like cellulase enzyme and protease enzyme break glycosidic bond in this part, for example, glycosidic bond, and the peptide bond that join successive amino acids in protein found in cell wall. Okay, look at the basic unit of beta glucose when joined together. Here look at O, join 1 and 5, carbon number 1 and 5. Here this O join carbon number 1 and 5 also. How glycosidic bond is formed? By rotation of beta glucose unit 180 degrees. This allow OH and OH beside each other, where condensation takes place and the glycosidic bond is formed by this manner. It's okay? Okay. Number 5. Question number 5. T lymphocytes are produced in bone marrow and mature in the thymus gland. When mature, T lymphocytes leave the thymus gland to travel throughout the body. They remain inactive inside organs such as the spleen and lymph nodes until activated by the presence of antigens. Figure 5.1 shows what happens to two inactive T lymphocytes, U1 and V1, in the presence of an antigen from a virus. U1 and V1 encountered by antigen, viral antigen. Okay, they become activated. U1 change into or activated into U2. After activation, it starts division, mitotic division, to form U3 and U4. The same happened to V1 after receiving antigen and complementary fatwa by its receptors with this viral antigen. It activated and changed into V2 that rapidly divide by mitosis into V3 and V4. This division to form clone expansion. This is called clone expansion. Okay, this is specific for certain antigen when complementary fit with receptors of T lymphocyte. It's okay, both U1 or V1. Here, for U3 and V3, cells derived from U3 remain in spleen for storage as reserve, lymph nodes, and circulation. Here, cells derived from V3 remain in spleen, lymph nodes as reserve, and circulation also. While U4, another group of cells from this clone expansion, okay, change into U4. U4 start to release of cell signaling molecules, chemicals for interaction, for communication with other white blood cells. Okay, cell signaling molecules. While V4 by itself attached to infected cells, try to distract it. Now look, U4 and V4 are types of activated T lymphocyte. T is the names given to these types of T lymphocyte. U4, V4. This U4, as it release of cell signaling molecules, that's the job of what? T helper cell. Okay, T helper cell. While V4, as it directed towards viral infected cells and try to distract it by certain mechanisms. This is called killer T cell or T killer cells or cytotoxic T lymphocyte. Okay, cytotoxic T lymphocyte. So U4 T helper cell, V4 T killer cell. It's okay. Now for letter A. Describes the roles of cells U4 and V4 in primary immune response. What's their role in primary immune response? Primary immune response means the encountering or facing the pathogen with its antigen for the first time, like during infection for the first time of certain pathogen, also during vaccination. Primary immune response due to vaccination. Here describes the roles of cells U4 and V4 in primary immune response. Now let's start with T helper cell. U4 represent T helper. Look at T helper cell in this short video. T helper cell secretes signaling molecules, cell signaling molecules called cytokines or lymphokines. This lymphokines activates other white blood cells like phagocytes and enhances phagocytosis. How come? Also trigger P lymphocytes to be at site of infection. Look at this cytokines and help B lymphocyte to divide, start to be activated and divide. Okay, and clone expansion takes place. Some of cells become plasma cells and other called memory. This plasma cells secrete antibodies in blue structure. This adhere or attach and they make clumping of pathogens. This enhances phagocytosis. Look at phagocyte. Now instead of eating or not eating, engulfing one bacterium, it engulfs a lot. Instead of engulfing one virus, it engulfs a lot of viruses clumping together by action of antibodies. Okay, now for U4, look, U4 releases cytokines to stimulate B lymphocytes. Stimulate for what? For division, to clone expansion, to differentiate into plasma cells and memory cells. And also what happens? U4 stimulates macrophages for phagocytosis, proper phagocytosis, enhances phagocytosis. Okay, now let's discuss the role of what? V4. V4, T killer lymphocyte, T cytotoxic lymphocyte, T killer or natural killer cell. Look at this short video. T killer cell, cytotoxic T lymphocyte. Where? This one with its receptor. What its role in primary immune response? This is called cell-mediated immunity. The role of cytotoxic T cell, cell-mediated means face-to-face, cell-to-cell. The cell infected by certain viral DNA. Look, injection of DNA. The cell become infected now. Okay. Infected cells release certain protein, thus enhances T killer cells to do its job. How T killer cells recognize these infected cells? By certain protein found on the surface of infected cells. Here, this T killer cells start to produce certain, either one of the two mechanisms, either secrete perforin, this chemical, or hydrogen peroxide, H2O2, or hydrolytic enzymes, or all of them, towards the infected cells to destruct it completely. Or it starts to make genomic change of infected cells by activating certain genes that leads to apoptosis, programmed cell death. Look again, it's another mechanism, apoptosis. Here's the left one, T killer, the right one, infected cell. Look, here activate certain program called apoptosis to destruct the cell completely. Okay, so two mechanisms takes place. Here, V4 releases chemicals like perforin, like H2O2, certain hydrolytic enzymes to kill infected cells. This is the first mechanism. Second mechanism, perforin creates spores, okay, for enzyme entry, also for inducing apoptosis, programmed cell death. As spores created in the wall of certain infected cells, it disturb osmotic balance, so this allow bursting of the host cell, okay, as it is infected by viral genome. Okay, letter B. Polio is a highly infectious viral disease. The virus infects the nervous system of humans. The disease can cause total paralysis within hours and can be fatal. The Global Polio Eradication Initiative, GBEI, was started in 1988 by the World Health Organization, WHO. In 2022, polio had been successfully eradicated from most of the world. However, cases of the disease have been recorded in some countries. Discuss the steps that must be taken by health authorities during vaccination program if an infectious disease such as polio is to be eradicated from the whole world. Here we in this question we asked about vaccination program, how it is done against polio virus. Of sure we have to make in our consideration, okay, or by health authorities take in their consideration, it provide this vaccine freely for the countries, for populations, especially poor countries. Okay, so the first step here, provide free vaccines globally throughout the world, okay, and create something called mass vaccination or herd vaccination. This is by help of volunteers in each country. Look, ensure herd immunity by vaccinating a large proportion. This herd immunity, it's type of active artificial immunity or vaccination. Herd immunity by vaccinating large proportion. Most of the population must be vaccinated except very elderly people, say patients with cancers, certain types of all types of cancers must be avoid this vaccination. Newly born capita or people, okay, all of them must be avoid vaccination, but most of the population must be vaccinated. It's okay. Ensure herd immunity by vaccinating large proportion of population. Third one, maintain accurate vaccination records about percentage of the population vaccinated already or not yet, and so on. Educate people about vaccines benefit and counter misinformation. Sometimes misinformation leads to avoid vaccinate our children. How come? Certain misinformation, misleading information through social media, for example, that relate autism to vaccines, certain type of vaccines. This make some parents avoid vaccinate their children, okay, fearing from autism, for example. This misinformation, not true, not scientific. There is no relation between autism and vaccination program or vaccines in general. Now, for number 6, letter A, student constructed a table to compare the structural features of plant cell, prokaryotic cell, and a virus. Complete table 6.1. Here, feature and comparing between plant cell, prokaryotic cell, and virus. As a point of view, feature external structure, size of ribosome, nucleic acids. For the first row, what about external feature, external structure of plant cell? Cell wall composed of cellulose. Prokaryotic cell also surrounded by cell wall but composed of murine or peptidoglycan. Okay, murine or peptidoglycan. Virus, no cell wall, but it is surrounded by protein coat. Capsid composed of capsomers or protein. Okay, protein capsid, protein capsomers. Here, what about size of ribosomes? For plant cell, 80S4 ribosomes found on the plasmic, rough endoplasmic reticulum or free in cytoplasm, and 70S ribosomes found in chloroplast, and the mitochondria also. Prokaryotic cell, the type of ribosome, the size of ribosome is 70S, like found in chloroplast and mitochondria. 70S ribosomes. Virus possess or have no organelles, even ribosomes. no ribosomes, no nucleus, no endoplasmic reticulum, no Golgi apparatus, nothing. Okay, it's a cellular. Nucleic acids for plant cell, either DNA and RNA. DNA nuclear and RNA as message needed for protein synthesis. Prokaryotic cell, DNA and RNA. Okay, virus, either DNA or RNA. DNA or RNA as genetic material, as nucleic acid processed by certain type of virus. Now for B, letter B, the cholera bacterium releases a protein toxin called chlorogen. The toxin causes the loss of chloride ions and water from epithelial cells into the lumen of intestine. Here's the lumen of intestine. This part, cell membrane. Okay, phospholipid bilayer of cell membrane of lining of intestine. Okay, when chlorogen binds to membrane of one of the cells, look at chlorogen, complex structure. Here, carbohydrate part joined with protein, this is called glycoprotein. When chlorogen fit, complementary fit with its receptor on cell surface membrane of intestine wall, here what happen, certain changes takes place. Binding causes endocytosis of chlorogen, yes. Then chlorogen as pathogen stimulate exit of chloride ions to outside to the lumen, okay, followed by water, okay, exit of chloride ions and followed by water. Now the question: state one likely source of an outbreak of cholera. What's the cause of outbreak of cholera? Contaminated water or food? Drinking, swimming, bathing, okay, in contaminated water, or even eating food, vegetables, fruits, okay, washed by contaminated water with sewage water. Here Vibrio cholerae, the causative agent, the pathogen, okay. It has one flagellum at one extremity and it's coma-like in shape, Vibrio cholerae. Okay, now double I, with reference to figure 6.1, state why chlorogen molecules are described as having quaternary structure. It is protein. Quaternary structure means it made of protein. Okay, now mechanism of action of cholera toxin. Chlorogen here, look, it is formed from many units of peptide that form protein, polypeptide chains. Okay, different polypeptide chains. Now chlorogen has multiple polypeptide chains, not one. Quaternary means more than one polypeptide chain joined together. Okay, chlorogen has multiple polypeptide chains, so it have quaternary structure. Now look, state the type of cell membrane component that forms the receptor for chlorogen. Look at the receptor here, this part. What its name? Type of cell membrane component. What's the chemical name of this component? Glyco means carbohydrate part joined with protein, so this is called glycoprotein receptor. Okay, glycoprotein receptor. Now for letter IV, the process by which chloride ions leave the epithelial cells requires energy. Name the phosphorylated nucleotide that is needed for this process. Nucleotide, but phosphorylated with two extra phosphate group. This is called ATP. Okay, ATP, adenosine triphosphate. Here only the nucleotide, but not phosphorylated. What about this one? Extra two phosphate groups. so it is called adenosine triphosphate. Now for V, explain why water also moves from epithelial cells into the lumen of the intestine. Okay. Letter V, explain why water also moves from epithelial cells into the lumen of the intestine when collagen is present. Look. Here the mechanism of releasing or loss of water and mineral from the intestine when subjected to collagen. Here the lumen of the intestine where bacteria found, cholera bacteria, secrete collagen. Collagen make disturbance of cell membrane of the wall of alimentary canal, intestinal wall, release chloride ions to outside to the lumen. This create low water potential inside the lumen, where the cell itself become with high water potential. So water move to outside also. Okay, so watery feces here in the lumen. This causes diarrhea, severe diarrhea. This water compensated from what? From blood. So blood also loss or lose its water content to the lumen to form watery feces. Okay, now explain why water also moves. Chloride ions lower the water potential into the lumen. Water moves into the lumen by osmosis. Look at this figure. Okay, here what happens? Here lower water potential in the lumen due to release or loss of chloride ions, and higher water potential inside the cells. This moves to outside to the lumen also, okay, by osmosis through aquaporins and through the cell membrane in any area. Thank you for watching. See you next time, inshallah.